welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And this was chapter Biotechnology and its applications. And we are in part 3 of the series. So if you haven't checked out part 1 and 2, please do so. The links would be there in the description box below. Alright, so let's begin. Okay, so question number 1. Maximum application of animal cell culture technology today is in the production of, this appeared in the year 2003, A. Insulin, B. Interferons, C. Vaccines, and D. Edible proteins. Okay, so the maximum application of animal cell culture technology today is in the production of C. Vaccines. Okay, the answer is C. Let's go to the discussion. So now animal cell culture technology is the use of mammalian cells or avian cells. Okay, so these cell cultures are made using these cells. Okay, and using these cell cultures, we can produce vaccines. Okay, now for example, polio vaccine or measles vaccines are produced using animal cell culture technology. Okay, so the production of this or the invention of this animal cell culture technology and then the formation of vaccines have proven to be very helpful. So ideally, vaccine production is a maximum use of this animal cell culture technology. All right, so it's like mostly like a factual based question. Learn it as a fact. Okay, so if they ever ask you, you can answer this. All right, let's move on. Question number 2. The term humulin is used for, this appeared in the year 1999, A. Hydrolytic enzyme, powerful antibiotic, C. Human insulin, D. isoenzyme. Okay, so from the term itself, humulin is used for human insulin. Okay, human insulin. Answer is C. You're already aware of the story, right? I'm not going to repeat it again. So there's a company called Eli Lilly, and they have managed to produce a synthetic insulin where they have produced A chain separately and then B chain separately. Okay, and then they were combined using those disulfide bonds. Now this is human insulin. Okay, that is produced using DNA technology. All right, and this product is actually called humulin. Okay, just remember it this way. Okay, this product is actually called humulin. It's human insulin and it is used as a medicine today, and the company Eli Lilly was able to produce this, create this, I'm sorry. So yeah, I've already discussed this application before. You can go back and watch my previous videos too, so I'm not going to much detail over here, but to answer your question, the question was about humulin, right? So humulin is actually human insulin. That's all. Okay, so that was again a direct question. All right, let's move on. Okay, human insulin is called humulin. Just to repeat on what I just said. All right, question number 3. Hybridoma cells are, and this appeared in the year 1999, A. Only cells having oncogenes, B. Product of spore formation bacteria, C. Nervous cells of frog, D. Hybrid cells resulting from myeloma cells. So the answer is D. Okay, hybridoma cells are hybrid cells resulting from myeloma cells. The answer is D, but it isn't very accurate. We'll see what is this hybridization coming from. Okay, so it's actually hybridoma cells are a fusion of spleen cells and myeloma cells. Okay, they're malignant plasma cells that are obtained from the bone marrow. So now the whole idea was to now why spleen cells are used, right? So the whole idea was to create a single line of antibodies, like a particular antibody for a particular antigen. We know when we have serum, we have lots of antibodies for different antigens. We don't want that. So let's have a cell line culture which produces only one type of antigen, antibody for a particular antigen. So how do they do that? Okay, so first for that you need the antibody, right? So mice were injected with the antigen. Okay, and so the mice would create antibodies for that particular antigen. Okay, and that was obtained from the spleen cells. Okay, so they took out the spleen cells which contain these antibodies. Now, okay, we've got the antibody, but how do we preserve it? How do we maintain the line of this antibody? We need something else too, right? So they used myeloma cells. Now myeloma cells are a type of malignant plasma cell. That means they have cancerous property. You know, right? Cancerous cells keep reproducing over and over again, right? They have that ability to reproduce over and over again. Okay, so now we have the antibody for the particular antigen. Now we are using the property of this malignant plasma cells, the myeloma cells. Okay, so what they did is, say these were the myeloma cells and they have the property of reproducing over and over again, and they fused this antibody into this, let's say, antibody into these cells. Okay, so now let's say if a few cells have that antibody, they will keep reproducing over and over again. Now you have a whole cell line culture of only that particular antibody. Okay, so now this becomes very useful in maintaining a cell line of a particular antibody. So whenever you want that antibody, you can use it. So this is actually called monoclonal antibody. This is one of the applications of hybridoma technology. Okay, so what I've just described is actually hybridoma technology. Okay, but the question says hybridoma cells, but this is basically what these hybridoma cells are. So what I've just described over here, this whole process is called hybridoma technology. Okay, so eventually you are producing a cell line culture that contains only one type of antibody which you want for a particular antigen, which is fair. Okay, and this eventually becomes useful for that particular antigen. To answer a question, yes, that statement pretty much answers the question. Hybrid cells, statement that I had given in the discussion, hybrid cells resulting from myeloma cells. But hybridization with what? That is not very clearly mentioned in this option here. So that's why I've explained it in detail in the discussion over here. All right. So basically, this is what this hybridoma cells and hybridoma technology does. Okay, let's move on then. Question number 4. Maximum number of existing transgenic animals is of, and this appeared in the year 2011. Okay, so the maximum number of existing transgenic animals is of A, fish, B, mice, C, cow, and D, pig. Okay, so the answer is actually mice. And I believe this answer is given in NCERT also. So 95% of transgenic animals are mice, and they developed to test the safety of vaccines. Not only that, they are also sometimes used for understanding, regulation of genes or testing, a product, or studying diseases. Okay. So, but to answer your question, yes, 95% of transgenic animals are mice. Okay. So the question says maximum number. So it's mice. Okay. Let's move on. Question number 5. Read the following 4 statements A to D about certain mistakes in 2 of them. Okay. So to find the mistakes. A. The first transgenic buffalo Rosie produced milk which was human alpha lactalbumin enriched. So Rosie was actually a transgenic cow. B. Restriction enzymes are used in isolation of DNA from other macromolecules. That's not really true. C. Downstream processing and other steps of rDNA technology. Yes. D. Disarmed pathogen vectors are also used in transfer of rDNA into the host. That's also true. So I think A and B are the mistakes. So the option should be D. This appeared in mains 2011. Answer is D. Okay, yeah. So A, in 97, Rosie, a transgenic cow, it wasn't a buffalo, produced human protein rich milk, alpha lactalbumin, and was balanced for human babies. So it provided as a food for human babies. Okay, and B, restriction enzymes, we all know what restriction enzymes do. Okay, so they are like molecular scissors and they isolate a fragment of DNA that codes for a palindromic sequence from a larger piece of DNA. We've already discussed restriction enzyme multiple times. And yeah, C and D were correct. Option C and D, so those were not mistakes. A and B were the mistakes, and that's basically the right answer. Okay, well, that's so simple. All right, so question number 6. Genetic engineering has been successfully used for producing, this appeared in the year 2010, A, transgenic mice for testing safety of polio vaccine before use in humans. B, transgenic models for studying new treatments for certain cardiac diseases. C, transgenic cow Rosie, which produces high fat milk for making ghee. D, animals like bulls for farm work as they have superpower. Okay, so genetic engineering has been successfully used for A, transgenic mice for testing safety of polio vaccine before using humans. We already discussed that, right? Previously. So they are 95% of the transgenic animals are mice, and they are one of the uses are that they are used for testing the safety of vaccines. Okay, so the answer is A. Yeah, we'll discuss this before, so I'm not going to go into detail. Yes, we can move on. Okay, so question number 7. Production of a human protein in bacteria by genetic engineering is possible because, and this appeared in the year 2005. A, the human chromosome can replicate in bacterial cell. D, the mechanism of gene regulation is identical in humans and bacteria. C, bacterial cell can carry out the RNA splicing reactions. D, the genetic code is universal. So how can we produce a human protein in bacteria? It's simple, because the genetic code is universal. All right, the genetic code is universal. So the answer is D. Let's go to the discussion. So we know that the bases A, T, G, and C, okay, they form codons, right? A, T, G, and C form these 3 base codons. I'm just giving examples. I don't know which codon translates to which protein. Okay, they will eventually translate into proteins, right? You know the dogma, right? The central dogma, that you have a DNA strand which then transcribes to mRNA, and then it eventually translates to protein. Okay, this is very important, the central dogma. Okay, so if this didn't exist, we wouldn't be able to form proteins. Okay, eventually we wouldn't be able to form proteins, which is our final goal. Okay, now this is universal. Okay, this ATGC, the central dogma is all universal. So if this was universal, we could do anything, right? We could create human proteins in bacteria, because bacteria just behaves like a host eventually. Okay, so bacteria behaves like a host. So irrespective of whether the human protein is formed in bacteria, can still produce a protein because the codons translate to that protein and the bacteria behaves like a host. So this is one important feature in nature. Okay, just think of it as this way: the nature has created this central dogma, or this general code, okay, that exists and that can, translate and form codons, different combinations of the bases, and then eventually translate into protein. Okay, so yeah, this eventually helps us understand why human protein in bacteria can be produced by genetic engineering. Okay, so we've used this idea to basically be able to produce proteins, human proteins in large numbers using bacteria. Okay, So genetic engineering is basically a man-made concept. So we've taken nature's model and we have created, or man has created another model or a man-made construct or a concept called genetic engineering, where we use this central dogma and then produce the target or whatever we want, the proteins that we want. Okay, clear? Let's move on. Question number 8. In transgenics, expression of transgene in target tissues determined by somewhere in the year 2004. So the expression of transgene is determined by A enhancer, B transgene, C promoter, D reporter. So this is a little complicated. You need to understand a lot of genetics in this. But to answer your question, the expression of transgene in target tissues is determined by D reporter. Okay, answer is D. Oops. Okay, so reporter genes are genes attached to the promoter sequence. So when you look at a vector, you know what promoter sequence is, right? I think we discussed that in the first part, biotechnology principles and processes. Okay, so it's like a promoter genes are present in the DNA. They contain the foreign gene and that needs to be translated into proteins. So basically it helps in initiating the transcription of gene. But when the initiation of transcription of gene is happening, we need to see if it has successfully happened or not, right? How do we know that the transcription has happened? That DNA is formed into mRNA is finally translated into proteins, but how do we test that? So there's something called reporter genes that are present, and from the name itself, you understand what are reporter genes. Reporter genes are basically reporting to you that yes, the translation has happened, or no, the translation has not happened, based on some assays that are done. Okay, some assays are done, and this helps. Let's say this is the reporter gene, and you have another gene called a promoter gene. Okay, the promoter gene's job is to initiate the transcription. Let's say if this does not initiate any transcription, so the reporter gene will not report anything. So we know that something has not worked out. Okay, that means the transcription has not been initiated, sorry. But let's say if the transcription has been initiated, eventually you form the mRNA and then eventually you get your proteins. It's like a signal sent to us. Okay, it's like a signal sent to us, okay, that yes, the transcription has happened. Okay, so reporter gene, if we go back to our question, the expression of transgene in target tissue is determined by reporter. Okay, think of it as this way. So this is a little tricky, but just understand the difference between promoter and reporter genes. Okay, then it'll be easier for you to understand this question and answer it if it ever appears in the exam. Okay, so this was actually a good question, a little tricky, but good to know. All right, let's move on. Okay, so question number 9. The transgenic animals are those which have, and this appeared in the year 95, A, foreign RNA in all its cells, B, foreign DNA in some of its cells, C, foreign DNA in all its cells, D, both A and B. Okay, so transgenic animals are those which have foreign DNA in all its cells. Okay, so the answer is C. So the DNA, basically transgenic animals are where the DNA have been manipulated. So they contain an extra foreign gene in them. So basically at the embryonic stem cell stage, there itself only, we will insert the foreign gene. So when the animal grows, let's say we're looking at mice. Okay, when the animal grows, or the mice grow up, they will contain the property of this foreign gene in all their cells. Okay, because it has been manipulated at the embryonic stage itself. Okay, so that is how transgenic animals have that DNA in all its cells. These animals are then used to study physiology, diseases, vaccine safety, chemical safety, produce biological products, whatever the case is, whatever you're trying to study. But this is how transgenic animals are produced or created. Okay, that they have foreign DNA in all its cells. Okay, this was a basic understanding. Yeah. Okay, question number 10. A new variety of rice was patented by a foreign company, though such varieties have been present in India for a long time. This is related to, this appeared in the year NEET 2018. A CO667, B Sharbati Sonora, C Lerma Rojo, and D Basmati. Okay, so a new variety of rice which was patented, and this variety has been existing in India for a very long time. So that is actually basmati. Is related to basmati. Answer is D. So the story goes is that an American company decided to, you know, create a cross or a hybrid between Indian basmati rice and semi-dwarf variety. Now, what they called this as a new product. Okay, they claim that this is a new product, and they successfully got the patent from the US Patent and Trademark Office. So once you receive a patent for this product, it calls for restrictions to other companies that they cannot sell this. But it is not a novel product because they have used the Indian basmati rice, okay, as one of that varieties for crossing it with the semi-dwarf variety. Okay, and Indian basmati rice is something that has existed for years and years in India. So obviously this was not fair to the farmers that were growing basmati rice and selling them, okay, as a means of their income. And obviously the Indian economy also would be affected. So eventually a case was filed, and I guess they won that in court. But this is what biopiracy is. Okay, even I think turmeric is also a product that is very indigenous to India. Even neem. So many companies, as in European or American companies, have tried to take this turmeric and neem and create novel products, so-called novel products, but it's not novel. It's not new because such things have existed in other countries for years. So it's not something that they can patent and then call for restrictions and restrict the selling of these products in other countries. So that's what the story is here. And a new variety of rice. The new variety of rice was the cross between Indian basmati rice and semi-dwarf rice. Okay, and of course such varieties have been present in India for a long time. So there are like about 27 types of basmati rice that are already existing in India. So it's not a novel product that they have created. Okay, so yes, that's related to basmati, to answer your question, and this is there in NCERT also, this particular example. Okay, so I would suggest you read it. All right, let's move on. Question number 11. In India, the organization responsible for assessing the safety of introducing genetically modified organisms for public uses, this appeared in NEET 2018. A. Indian Council of Medical Research, Council for Scientific and Industrial Research, C. Research Committee on Genetic Manipulation, D. Genetic Engineering Appraisal Committee. So it's actually GEAC, which is an Indian organization, and they assess the safety of introducing genetically modified organisms for public use. So the answer is D. Okay, so this organization makes decisions regarding the validity of GM research and safety of introducing GM organisms for public service. All right, so this is again present in NCERT, so you can go back and read the definition and what this committee actually does. Okay, so yep, this was pretty direct. Move on. Question number 12. Use of bioresources by multinational companies and organizations without authorization from the concerned country and its people is called, Sapirani 2018, A. Bioinfringement, B. Biopiracy, C. Biodegradation, D. Bioexploitation. So the answer is B. Biopiracy. Just like I gave you the example of the basmati rice, That's an example of biopiracy. So what they do is this, it fits the definition over here. So okay, refers to the use of bioresources by multinational companies, which was that American company, without proper authorization from the countries and the people concerned, without compensatory payment. So that American company used the basmati rice, which was very indigenous to the Indian continent, subcontinent, that country, and they did not provide compensatory payment. They did not take proper authorization. They just did what they wanted to do. But obviously, this comes under the category of biopiracy. All right. Yep. So basically, this is the definition. Again, this is also mentioned in NCRT. So you can go back and read it. All right. Let's move on. Okay, so question number 13. Which body of the Government of India regulates GM research and safety of introducing GM organisms for public services? It's appeared in 2015, cancelled exam. Okay, I think we discussed this before. A. Genetic Engineering Approval Committee. B. Research Committee on Genetic Manipulation. C. Biosafety Committee. D. Indian Council of Agricultural Research. So it is GEAC. The answer is A. So Indian government has set up organizations such as GEAC, which will make decisions regarding the validity of GM research and the safety of introducing GM organisms for public services. Okay, so this was just a repeat of the previous question. Okay, so we come to the end of this video lecture, and I hope you've enjoyed today's video lecture, and I hope you learned something new today. Please don't forget to like, share, and subscribe to MG NEET Biology, and I'm available on Facebook, Twitter, Instagram, so you can follow me there for more updates, and I'll see you in the next video lecture. All right, so bye.